And so in this case, right, so I might have two groups, so maybe that's useful or interesting to me. Whatever, this is just a cartoon example. And then we have supervised learning, which relies on labels on data. So I've gotten all these mushrooms and I've measured all these things about them. But I might also attach a label to that. So maybe I'm thinking about, oh, well, I want to know if these mushrooms are toxic or non-toxic. Okay, well, I have to do some experimentation or something to try to get that label. But then I have an extra dimension here represented by color. Some of these mushrooms are toxic, some of them are non-toxic. And then I would say a training sample, so not just a sample, but a training sample is a set of examples that also have labels associated with them. And then in supervised learning, the idea is we're going to take labeled data and we're going to build some function, a classifier, that can discriminate between the labels of data, right? So if I draw a boundary here, I might say like, oh, well, the blue ones are on this side, or the toxic ones are on this side, and the non-toxic ones are on this side, right? So that in the future, if I go out in the forest and I grab a mushroom I don't recognize, I can measure its height and cap width, and I can plop it into my classifier space here, and it looks like, oh, well, okay, if I want to see it or guess if this is toxic or non-toxic, I just see which side of the line it lies on, right? And in this case, it's on the orange side, so let's just say that's the non-toxic side. So presumably I can eat that mushroom. I'm probably not going to do that because I'm a little bit risk averse, but whatever, I could if I wanted to. But now that I've done this, let's say I also go out and I say like, well, I don't know if I'm like super confident in my model. I'm just going to collect a whole bunch more mushrooms, and I'm not going to spend all the timely process of like getting labels for those mushrooms, but maybe I can use the distribution of the data to inform my model to make it better. So in this case, again, I go out in the forest, I collect a bunch more mushrooms, and I measure their height and cap width, and I plot them in the space. And to the human eye, I might say, boy, it sure looks to me like my classifier isn't right. Like it just doesn't look quite right. You can see that. And so semi-supervised learning is basically using a combination of labeled and unlabeled data to try to come up with a better classifier. So that might be my better line. So this time, if I take that mushroom that I was looking at before, oh, now maybe it's actually toxic. So maybe I shouldn't eat it, right? Okay, so that's like the high level overview of machine learning algorithms. And I'm going to go a little bit more in depth on supervised learning algorithms because I think that's the most common thing that people are looking at typically when you're, in a lot of biomedical literature. But maybe if there are any questions, I'll give it a few seconds. Hearing none, I'm going to proceed and just give you a few examples of different algorithms, different supervised learning algorithms. And I'm not going to again go into great depth, but I'll just give you an idea. So, oh, right. And one might describe machine learning algorithms as being a combination of representation, objective, and optimization, where the representation is like, how does the model represent the space of data, and what decisions can it make about the data? So like, what is the space of possible models that it can come up with? The objective is basically an internal function that tells the algorithm what is a good model. So it's a measurement of like how do we improve our model, and then optimization is like the step-by-step process, the internal like algorithm that we use to adjust our current model until we get to like a good fit, till we get to something that is a higher value for the objective function within our representation. Okay, so I'll just go back to this set of these mushrooms again. I have toxic mushrooms and non-toxic mushrooms, and I came up with this dividing line between these in this two-dimensional space. But how would I do that without a human just showing up and like drawing a line and being like, oh, there's my dividing line, right? Like that might be a natural thing that a human would do, but it's not obvious necessarily how that would work within a computer. But the idea here is that for support vector machines, we make a decision to say like, we're going to come up with something that finds lines that have a large margin between the line and the nearest points. So there could be several different options for where we would draw that line, right? But this line, so my objective function is finding these large margins. This particular line, this dividing line, has a relatively large but maybe kind of a small, I don't know, like it has a margin, but if I compare it to this one has a larger margin between the dividing line and the nearest points, right? So this one is the better one. And I'm not going to go into like the optimization of how this works, but basically you can imagine like you're going to twiddle around with this line until you find the maximum margin. So that's one way, one algorithm that just the representation is a dividing line or plane in the space. The objective is to maximize the margin of that line, so its distance from the nearest positive and negative labeled examples. And there's an optimization step that wiggles it around to find the one that has the maximum margin. But support vector machines do have ways of accounting for things lying on the wrong side in your training data, but here is a clear example where I can't just come up with a single line that divides this space, right? That just divides it cleanly, at least. It's just not possible, not a single straight line, at least. So let's talk about a different type of algorithm. So here, I'm going to talk about decision trees. And these use a very different representation of the space. So they're not just finding some line that divides the space in two and then says one side is this label and the other side is this label, but rather it allows me to, the idea is it's going to use rules basically to divide the space up into chunks, and then the maximum or the higher proportion label in that chunk is the label for any new examples that show up in there. So what does that mean exactly? And the way it's going to do that is it's going to guide itself by information theory. So it's like entropy. So we have roughly equal numbers of blue and orange circles in this space. So here's just a rough, this block here is just a rough representation of blue and orange proportions. If I don't divide up the space, it's about 50-50. Now, that's not useful because if I randomly draw some new example, right, like that I have no information, 50-50. I don't know if it's going to be positive or negative. So instead, we're gonna pick one of our features and we're going to ask something about it. So we're going to take cap width, for example, and we'll say, okay, well, what if I divide the space such that one part of the space has cap width less than 1.25, and then the other part of the space is cap width greater than or equal to 1.25. Okay, well, we can see here that when I divide the examples up into that space, I get better information. So in this bucket, there are more blue than there are orange. So if I were to randomly draw a sample in that space, I would have a better chance of guessing correctly that it is blue versus orange. And the opposite is true for the other side. And what that looks like is I've first drawn a line in the space at cap width 1.25, so roughly halfway in this space. Okay. Next thing I can do is I can then take one of those other spaces and subdivide it once more. So now I might take that left space and I might divide it by height. And maybe I say, okay, well, height's less than 1.75 go over here. Height's greater than 1.75 go over here. And now I can see right in my leaf of this little tree that I'm building here, the one on the right here, so with cap width less than 1.25 and height greater than 1.75, they're all blue. So if I were to randomly draw in that space, it seems likely that I would have a blue example. And again, similar proportion, but like useful information in the left bucket. And so it looks like this now with that line in there. Then I can ask another one. Maybe I do height in this other bucket now, in this other space. Now I can choose a different splitting point for height there, right? So like height less than 1.5 and height greater than or equal to 1.5. So I have pure buckets or whatever on that side, which looks like that, say. So now maybe I say like, oh, well, if I draw one in this space here with these orange examples, it's probably orange. And then I could do it once more. I could subdivide that final space even further by height. So cap width less than 1.25, height less than 1.75, and then divide the height a little bit further, less than 1.3 and greater than or equal to 1.3. I now have, again, like pure buckets. So that's kind of the idea, is you iterate through your features, you find dividing points that improve some sort of criterion, like the purity of the entropy. We won't get into information theory, but you can use these types of measures to try to improve your ability to discriminate things into different buckets. So that's, it's also useful, I guess, in one way. I'll just, before we move on, decision trees are useful because they're interpretable, right? So like, it allows me to actually like look at this thing. I can sort of interpret the support vector machine by being like, oh, well, the thing lies on this side of the line, so I think it's this label, right? But that's not super useful, and I can't really use that to understand anything about the measurements that I've taken. But in this case, I can say like maybe some expert in mushrooms, and this is not real data, but some expert in mushrooms might be like, oh, if the cap width is greater than 1.25 and the height is less than 1.5, it's safe to eat or something like that. Maybe there's some intuitive reason why that might be the case. Like, oh, maybe you discover some new knowledge based on dividing the space up by these features. So that's like one of the ways you can look at this too, is these types of models are interpretable as well.
Hey, Finn, there's a
Yeah,
question in the chat from Renee.
yeah.
It's, is there a limit to how many dimensions? And I don't know if it means if that is equal to features or not, but to dimensions this machine learning approach can handle.
Yeah, so that's a good question. I tried to like avoid this. So in this case, like in the examples I'm using, yeah, the dimensionality is equal to the number of features that you've measured. And well, that's only technically true in support vector machines. But so the short answer is no, there's no like real limit to the number of dimensions you can measure. And I don't think I even talked about this. Maybe I briefly touched on this last time, but there are practical limits. So there are a few different things you want to consider when you like. It's sometimes useful to add more data, like more measurements or something. But the problem with just adding more and more is that you get into what's called the curse of dimensionality. So basically, any one feature could be washed out. Like some useful feature could be washed out by all the other features that are maybe meaningless, or maybe they're meaningful. But it's just the how do I say, the importance of any one feature is just less, right? And you also get into situations where like some features might be really highly correlated, and that can be sometimes not useful. Like if you're doing like some sort of statistical model, you might find correlations where there aren't, or you might, there's lots more chance for just spurious stuff. But the short answer is like no. Technically, there isn't really a limit to the number of features you could incorporate into one of these models. Again, it's I don't know if I'm really answering the question. Hopefully that sort of answers it. But if you want to ask about it more, feel free to come off mic and we can chat about it a little bit. But
Yeah,
go ahead.
I was thinking about like even with single cell data, how there are like thousands of uh, genes and etc. that are leading to this many dimensional space. But so is that why when we're analyzing it, you're kind of limiting to so many kind of principal components that you kind of limit the most influential factors then to try to limit that curse you're talking about?
Yeah, potentially. So like a principal component analysis is useful also just for visualizing, right? Because you find like it's sort of like you have this really high dimensional box that you're looking into, right? And there's data in that box, and maybe you have good sampled spaces within the box here and there and so on and so forth. But someone else has described PCA as being like rotating the box and looking at it at a more meaningful direction, right? So like you're capturing more of like what are the angles of looking at this box of information that just tell you more about the space. So I think you frequently, yeah, you frequently want to do dimensionality reduction in those cases because there are so many opportunities for spurious correlations between genes and stuff, right? Like whether I guess they could be spurious, or they could just be like one gene is like directly downstream from another or something like that, right? Like, or you're using, I'm not answering this well, sorry. The algorithms themselves don't require you, I guess, is the point, to downsample the space or to reduce the dimensionality in any way. But it is often useful for either visualization purposes or understanding. You might remove, I'll give you one example, right? So not PCA, but you might just remove a bunch of housekeeping genes, for example, like, so you can reduce the dimensionality that way. Or you might be particularly interested in just one pathway or something like that, right? So now you just eliminate everything except for the genes in this one pathway. There are a lot of different reasons you could want to reduce your dimensionality. One thing that maybe one concrete thing I would say is I wouldn't want to use something like a decision tree on a really, really high dimensional space because of this recursive dimensionality problem. Like, it's going to pick one feature at a time and it's going to split on that feature. And depending on how you set up your algorithm to like choose those features, it might be a super meaningful first decision, or it could be random. Like some algorithms are like, oh, well, the feature I'm going to split on this time, I'm just going to randomly draw it out of a bag. And if I have roughly 20,000, as for genes, for example, if I have roughly 20,000 features that I could select from, what is the chance that I'm going to pick a useful one to split on? It's going to split on it, but does that, if I ask the question, then like the interpretability thing, right? Like if I'm trying to use gene expression data to like predict something, now I have these rules and I'm like, if BMP4 is greater than 1,000 TPM and I don't know, Notch is less than 5,000 TPM and 4 other genes, blah, blah, blah. That's not a super useful model for interpretation. And you probably won't be accurate either. But like one of the big advantages of a thing like decision tree is like you can sort of read it like rules, and you're probably not getting something that's really interpretable at that point. I don't know. I'm just rambling at this point. Does that make sort of touching on what you're wondering?
Yep, thank you.
Okay, sorry. So short story long. There are lots of different reasons. It depends on the application too, but there's no technical reason. It's usually just based on how you're trying to approach the problem. Okay, and then I'll just give, I think this is my last example of a way of building a supervised learning model or a predictive model from labeled data, and that's dead simple. It's this k nearest neighbor model where the idea is I have some new point that arrives and I'm going to say that it's equal to its nearest neighbors, right? It's most like its neighbors. So if k is 1, this is a tunable parameter. If k is 1, I'm going to say it is the same label as its very near its single nearest neighbor. So in this case, k equals 1, it's 100% blue. If I say 2, I pick the second nearest neighbor. Now it's like, okay, it's like 50% blue. Okay, that's not super useful. Now I get the next nearest neighbor, and now we're talking about k equals 3. Okay, so here it's like 66% blue. Next neighbor, okay, it's like 75% blue. Maybe 4 is the right number, who knows? And you could like weight this by distances and stuff like that. But k nearest neighbor is like this dead simple model where you plop a new example into the space and you just say it is proportionally, or it is it's like the majority of its neighbors, basically. Okay. Any other questions about, so that's just giving you a taste of like the types of algorithms and like kind of how they work. And the way there's like bazillion different algorithms you can choose from. And choosing an algorithm is like an art in itself and also like a science in itself. It's, depends on your problem, depends on what your goals are. If your goals are interpretability, you already narrow your set of potential algorithms. If you just care about, accuracy or something like that, you might be looking at a totally different set of models that are much more complex. You might be looking at a balance of those things. There's all sorts of ways you could approach that, but they all kind of work the same way. You have some representation, you have some objective that they're trying to meet, and then you have some optimization steps, some iterative process that they use to improve their objective. Okay. And then, so let's say if they're, I'll let people come off mic if they have questions, but let's say now I've chosen my my algorithm, or no, let's say I want to pick between, I don't know, 3 algorithms or something like that, right? I've chosen some subset of algorithms. How do I decide which one is the best one? Or how do I decide if I should go with a model? Like how good is this model, right? That was another question that people came up with, is how do you evaluate these models? Well, the ideal testing regime, or at least historically how it's been taught, and maybe this is kind of changing actually, but the ideal approach would be you have this big old set of training data, more the merrier, right? Typically, Not always necessary, but it's often great. You use the training data, you put it through an algorithm, and it produces, I'm pointing at my screen rather than use my cursor. You have this training data, you pump it through an algorithm, and that produces some model, like a classifier, for example. And then you get a whole bunch more data, and you've never seen it before, you never touched it, and it's very similar to your training data. Same distributions, same places that you gathered it, same measurements, obviously. And then you push that new data through the model, and you say, did I get the correct answers? And that's how you would evaluate it, right? Like, and there are lots of different metrics you can use to evaluate it, but that's the idea. It's like you have training data, you train a model, and then you take all this new unseen data before and you put it through the model, and you see how well you did on that by some metric. That's usually not going to be possible, like for just several practical reasons, but like, you just have limited data. You can't do it. It's so rare. Maybe if you're like generating data from some mathematical function or something like that, you can do that. But most of the time, you're not going to be able to do that in the real world. So what we end up actually doing is something called cross-validation, where we have this data set, and instead of just training on it and evaluating on it, because that's going to be a biased measure, the model has seen the training data before when you're fitting a model, right? So you don't want to test it on the training data because who knows, maybe the model, think of the K nearest neighbor. If you train your model on your training set and then you test it on your training set, one nearest neighbor is going to be a perfect model because it will literally be its closest neighbor will be itself. So every example in your test set at that point will just sit right on top of a point in your training data set. So not super useful. So what you want to do is you break your data set into chunks and you do it and you train a model several times. So basically I have taken this block of data and I've broken it into 5 chunks or folds in this case. And each time that I build a model, I'm going to build 5 different models. I'm going to hold one chunk aside for evaluation, and I'm going to train the model on the other stuff on 4 different folds, right? So that's all this is representing. Is like I'm going to train a model 4 to 5 times, and I'm going to test it on each successive chunk in each of those. There are lots of different ways you could do this, but that's kind of the idea. And then let's talk about actually evaluating a model, right? So just for some preliminary information here, right? Like I'm going to use the letter P to equal the number of positive examples and to equal the number of negative examples. True positives are the number of positives that were labeled positive by some classifier. And here I'm just looking at this as like a binary problem. Obviously, these things change when you look at like some sort of multi-label problem or when you're looking at a regression problem. There's only so much we can talk about in one go. I just want to give you the flavor, right? So true positives are is the number of positives labeled positive. False negatives are positive examples that were labeled negative, so they're falsely labeled negative. True negatives are negative examples that were labeled negative, so correctly labeled negative. And then false positives are negative examples that were labeled positive, right? There's a little box down here to sort of represent it, right? So you have labeled, actual, it's a confusion matrix. There are other terms for it, but yeah, that's the idea. So probably the most recognizable single measure, like the most intuitive or simplest single measure of evaluating a model when you have a positive and negative label, like a binary model, is just accuracy. So what proportion of all of my positive and negative examples did I correctly classify? So that's the true positives plus the true negatives over the entire positive plus negative, the entire size of the data set. Okay, now that's a useful measure. We think about accuracy probably all the time. Well, maybe, I don't know, maybe normal people don't, whatever. It's just like a standard thing. It's like the most intuitive thing that comes to mind when you think about evaluating if something is accurate, right? You just say like, how many of the things in the data set did it label correctly? But that's not super useful in many examples, right? So think about, and this is especially true, I think, of biomedicine. Think about really skewed data sets. So let's say 1% of your data set is positive. So maybe this is like a cancer, some like not super rare, but relatively rare disease or something like that, right? So cancer, 1%. Well, let's say cancer, whatever, that varies a lot. Some disease, 1% of your data set is positive. And now you built a model. So 99% is negative, 1% is positive. You built a model on it. and you measured the accuracy of your model and it said, wow, you got 99% accuracy. That sounds amazing. Is that good? Probably not, right? Because all it had to do was label everything as negative and it got 99% accuracy. Is that what it's doing? I don't know, maybe, but probably. But you don't know, right? That's the point. It's like skewed data sets, accuracy is going to be a terrible measure. Precision is one way of trying to get around that. So you can look instead just at your examples that your model labeled positive. What proportion of the examples that your model labeled positive are in fact positive? So if my model says something is positive, what's the probability that it's actually positive, right? And that might be useful for diagnostics of some sort, right? But that ignores things like, what's the probability that it gets negatives right? It just depends on the different costs of getting those things wrong. And it doesn't tell you, like, maybe it just labels very few things positive. So maybe it's like it picks its most confident one and it says that one's positive and it got it right. Ta-da, 100% precision. Great. So it doesn't take into account another thing like recall. So recall tries to account for that piece of information. So recall is just the proportion of your positive examples that your model captured in its prediction of positive. So did it get all of them? This is important for things like cancer predictions, right? You don't want to miss cancer. So I don't know, you're building some model and it's trying to predict cancer in some patient. You want to have really high recall, probably, I would guess. So you want your model to capture all of the cancers in that case. And then there's a false positive rate, which is, there's a lot more than this, but these are just really common ones. False positive rate is like your alarm rate. So like what proportion of your negative examples did your model accidentally label as positive? Because they're negative, that's also useful for diagnostic type criteria, right? Because there are negative side effects to incorrectly saying that something is bad when it's not. Any questions? I don't think I saw any in the chat, but okay. We'll move on then. So, all right, so these are single measures of that you can use to evaluate your models. But maybe we want to look more holistically at a model, because like so let's say I built some model, or I trained some model, and it gives me a probability out, right? So now I can give it any example in my test set, and it will say, what's the probability that that is positive? That means I can give a whole ranking. I can take an entire data set and put all my examples through it, and I can rank them by their probability that my model gave them as positive. Now I might now I want to choose a good threshold for my model. So it might not actually be a probability, but it can rank my examples, and I might want to choose what's the threshold at which my classifier actually says something is positive. Again, I have a ranking. Do I put it over here? Do I make it really conservative? Do I put it over here to make it like really open? Like, does it? That's just kind of the ideas. I want to make a ranking, and I can evaluate my model on that ranking. So one really common one, kind of dead simple, is the ROC, or the receiver operating characteristic. There's some interesting history behind it with radar or whatever. We won't talk about that. But the idea is basically it's balancing, the curve itself is balancing recall and the false alarm rate. So the best point of this curve in this space of ROC is this top left corner, which basically says if I can get an operating point up in this top left corner, that means I have perfect recall with no false positives. So no alarms going off, or no false alarms. And the way it kind of works, like visually, we might look at it like, here I have a model ranking. I apologize if this is actually an older slide. I have red green here. I hope no one is red green colorblind, but we've got pluses and minuses as well. So hopefully that helps. But the idea is every time that I correctly, and I'm saying that left is more likely to be positive, right is less likely to be positive. So each time that I move my classifier threshold, I tick it to the right through my examples, I'm capturing a new example. And in this case, like, okay, let's say I set my threshold right here for my classifier. That means I called one example positive and it was positive. Okay, so that's, great. There's zero false alarms, but the recall is kind of low. Okay, so let's loosen the threshold a little bit more. Go one more example over. Oh, well, that time actually I got a negative example. So I increased my false alarm without increasing my recall. Okay, let's go a little further. All right, so I go one more step. Oh, I got it again. I got another positive example. So actually, yeah, that's great. I'm increasing my recall and like not decreasing my false alarm rate, but like my precision is maybe a little higher at that point. But it's just like building this step function. So each time I encounter a positive example, I go up. Each time I encounter a negative example, I go right. And along the diagonal is sort of like the baseline 50-50. But as you can see on the right here, I just have like another example where I have more positive examples ranked more closely to the positive end. And that means I get a higher curve. So the area under the ROC curve is as you increase that, that's better. You can sort of interpret it as like, or you can definitely interpret it as, if I were to draw one random positive example and one negative example from my data set and put them in my algorithm or put them in my model, what is the probability that my model would rank the positive one as more likely to be positive than the negative one? So it's just a way to measure a more holistic view of how my model is doing. But it's not great on skewed data. Again, sort of runs into the same like problem as like measuring accuracy. So in that case, I might want to use like a precision recall curve. So where the best operating point is in the top right corner. But basically we have precision on the vertical axis and recall on the right. So on the far right, I will have, if I really loosen up my classifier and call everything, absolutely everything positive, I'll get 100% recall, but my precision will be bad. So we're just trying to improve our recall without decreasing our precision. But so I don't think I'll belabor this any further because we don't have a ton of time. We can talk about it more, but these are just two different ways to evaluate it. There's like a bazillion different ways to evaluate these models. But the most important thing for biomedical practice here is you always want to compare to the standard of care. Yeah, go ahead, Michael.
Is there any, will we learn how to actually do these things during this at all? Or is just like a kind of
how to like measure ROC and such?
I mean, like actually using a program to do our stats. Like, I know
yep.
For me, I do this stuff all the time. Not positive predictive value, negative predictive value. I just need to implement it.
Yeah. So not in this talk, but next time the plan is I think what I'd like to do and what we're planning to do is I'm just like literally I'm not gonna like explain in great depth like but I'm gonna have like code. We're gonna like go through code. I'm gonna have a dummy data set, or but maybe not totally dummy data set, but I'll have a data set and we'll go through step by step like some exploratory data analysis. We'll go through building a model, evaluating it, visualizing the results, and so on and so forth. So hopefully that's helpful. Do you think that'll sort of get at what you're looking for?
Yeah, what program? In R or what?
Oh, so right, that's another thing. So often in data science, you typically have Python people and R people, and then a combination of both. I will probably do most of it in Python. I can mostly because that's just I'm more of a Python guy. I can do it in R, but I would also probably make mistakes or do dumb things.
So if I have zero coding experience, will I be able to get those skills during this or not adequately?
I think you'll be able to get some of them. It's not going to be like an end all be all just because it's too big a field to learn that stuff. I can point you and others, like I can point people to resources to learn more in depth. Like we have like the Data Science Hub, for example, on campus has like, I don't know if you're familiar, but they have like regular classes basically they put on for like learning. It's usually particular skill sets, but you can look at what they're offering and you can find things like, oh, well, this time they're going to be talking about I don't know, like doing stats in Python or something like that, or it'll be like a PyTorch tutorial or something. What I will get to here is like the next thing I'm going to go through is just like some of the common tools that I use as like a more Python oriented data scientist. And it could be useful because you can say like, oh, well, that's what that tool is for. Now I can just like go to the documentation for that if there's good documentation, which isn't always the case. But at least it gives you like a mental model to work from, like what should I be looking at even to do some of this. So hopefully that's helpful. Yeah, right. But yeah, next time I hope to go a little bit more in depth. And please, do take the survey, everyone, afterward, and say like, what are the things you really want to know? And maybe in those examples that I go through next time, I can like be like, oh, well, EDA or exploratory data analysis is useful, but maybe not everyone like cares about this particular aspect. So we'll go a little bit more into certain aspects. So please do provide feedback at the end so that I can guide my preparation. But so just going back to the point here, right? There's a bazillion different ways you can evaluate these models, but especially in the biomedical space, when people's health is on the line, always compare to the standard of care. I've read so many papers where some computer scientists like myself, has like published some paper on some, I don't know, some biomedical data set of some sort, and they're like, oh, our accuracy is really high, or our ROC area is really high. It's like, yeah, but what does that matter? Do the doctors like that? Is it good for patients? Who knows? So like, you want to know like, how is treatment efficacy measured? And are you measuring that when you're evaluating your models? Is your data set representative of the real world? How bad is a false negative? How bad is a false positive? And what trade-offs are there, right? So you just always have to be thinking about the standard of care. So that's the most important thing. I don't know why that doesn't, it's just still a problem. I don't know. Okay, so some tools of the trade. So hopefully Michael, this helps a little bit, at least from the Python side, at least. I think, again, I was sort of touching on this. There's sort of like, in the data science world, Python and R are probably like the most common languages used. And like, it's not like there's a battle between them, but I think some people tend to be more Python people and some people tend to be more R people. Most people can go between them a little bit, but you definitely like, you have a first language or something like that in any case. But so, all right, so I'm more of a Python guy, so here's a bunch of tools of my trade. I always like to throw this in here as a joke. It's not a good joke, but pandas is a really popular tool. It's not actually pandas. And then we'll talk a little bit about NumPy, SciPy, Seaborn, Matplotlib, scikit-learn, PyTorch, and Request. And I'm just going to give you an idea of what each of these are, so that in the future, if you want to explore these more, you can look it up. So pandas is basically, I just call it Excel for Python. It allows you to read and write data in many different formats. Basically, it's a programmatic tool to allow you to like clean, filter, and reshape data. So you load in data and you can do some common math and stats functions as I note here, but the idea is like you're going to clean data and reshape it and such, probably with pandas. That's a really common tool to use for that stuff. And it works well with a lot of other packages. NumPy, also really common, has some similar vibe. You can read and write a lot of different, well, a handful of different data formats, but basically it's focused around matrix and list data structures. It allows you to do really fast vector math, so like matrix multiplication type stuff. It's super memory efficient, and you can like sample data and reshape data there too. And it's kind of like the underlying package of a lot of other packages or programming packages. I would just say pandas is a little bit more friendly for like exploratory data analysis than NumPy. SciPy, tightly coupled with NumPy, allows you to do a lot more like advanced stats and modeling. So there are a lot of built-in stats models. There are a lot of like stats algorithms, or like, or hypothesis testing, stuff like that. There's signal processing built in. Yeah, sure, Rachel.
Hi, sorry, you are welcome to finish, but I have a question just about if I'm looking to work with a lot of text data, what type of programs might be best optimized for text?
Yeah, that's a good question. I'm not going to be able to give you a good answer. Typically, what ends up happening is your text gets encoded as like numbers, basically. So that's typically what ends up happening internally, right? That's going to be the representation. And how that's done depends on what you're trying to do. If you're talking about like you want to like say you want to like classify documents or something like that, or the classic like toy example is like sentiment analysis from tweets or something like that, right? And maybe that's not the classic example, or maybe that's so old that I don't know. But typically the idea is you have these you're going to convert it into some other data format so that it works with some like predictive algorithm or something. But now there's all these, and I'll touch on this very briefly. There's all these like these large language models now that allow you to work with text much more cleanly. I guess maybe the answer is like almost certainly the answer these days would be like using large language models like OpenAI's GPT or something like that. But it really depends on what you want to do. So maybe we can talk offline or after at the end just about what you're trying to do, and we'll touch on. And I'll try to answer the question at least, but yeah, it depends.
Hey,
Um,
Finn, we are starting to run just a little close to time.
Yeah.
So I don't know if you want to open it up soon to broader
Sure.
questions.
Yeah. I only
Okay.
have a few more slides, so
Okay,
we should.
perfect. Thank you.
Seaborn and Matplotlib visualization tools. So make it really easy to make different figures. They're super customizable. Seaborn is a little bit more like their pre-baked figure types that you can generate. Matplotlib is a little bit more options, but like it's a little bit harder. Documentation is good for both. Scikit-learn is like broad machine learning tools. So lots of different types of algorithms and like evaluation and feature selection type algorithms. And there's a really great standardized API design and it's well documented. And then PyTorch, which is more in the deep learning world tool set. So this is like the new hotness, or not even that new anymore, but the hotness right now. Allows for like GPU acceleration, has like higher and lower level APIs, but it's all about deep learning. And it comes with like some pre-trained models baked in as well. And by pre-trained models, I mean things like foundation models. We talked about this, or this came up in conversation. What the heck is a foundation model? And again, I'll just touch on this as a bonus tool here. They're basically just general ML models on large data sets. So I had like lots and lots of text or lots of images. There are a whole bunch of different versions of these. There's like the OpenAI's like GPTs. Llama is one of Meta's foundation models, but they're like general models that are good at representing data. And then what you do is you build wrappers around it to do something with it. So like ChatGPT is a chat wrapper built around GPT. And again, we can talk more in detail, but that's kind of the idea. And these are like huge models, right? And then requests, which was on the first screen that I had there. Requests comes up a lot when you need to like pull data from a website. So it's good for like web scraping, or if there's some API that you need to hit on a website to capture data. Allows you to do that really easily. So right there, we've got 5 minutes at least. So please ask away any questions you may have. Sorry, there's a whirlwind.